Bismillahirrahmanirrahim. Thank you for the invitation. It's actually complex even for us breast oncologists, all of this new development. And one of the new development that happened in the field of breast oncology was introduction of CDK4/6 inhibitors. One of the strategy to overcome endocrine resistance, one of the most important obstacles that we are facing in treating our patients. As we know, ER-positive, HER2 negative disease, luminal A and luminal B subtypes, we think about them as a distinct type of breast cancer. It's distinct in the natural history of the disease, it's distinct in how we treat them, and also we think it's distinct in biology of the tumor itself. And as we having a lot of progress in treating our human breast cancer, and even improving the survival and curing most of the early stage luminal breast cancer, we still have a substantial rate of recurrence, even years and years after finishing adjuvant treatment. Sometimes it's extended to 10, 15 years, which is unique actually for this subtype of breast cancer. And the other challenge that we face is also how to control the disease when you have a drug that's effective, and to how to maintain this efficacy while maintaining the good quality of life. In 2017, we used a strategy and approach to sequence our treatment, hopefully to delay the chemotherapy and to maintain the quality of life of the patient. So using the most effective, less toxic treatment is the main thing that we are doing, especially for advanced and metastatic disease. And uncovering the molecular mechanisms and our understanding about molecular mechanism have stimulated research to introducing a new targeted treatment, and also even made the researcher look back into all their agents and reuse them in a different setting with a different dosage. And actually this brought some success in this field. Of course, in my mind, and I think everyone is agreeing about that, molecular and genetic profiling will be the next step in improving our care to our patients. Talking about endocrine resistance is actually one of the main obstacles that we are facing, and this for maybe most of the targeted treatment, and especially in endocrine therapy treating patients with advanced breast cancer. We are using an effective drug and blocking one pathway for some time. With the time, resistance will develop, and blocking this pathway will not be more effective, and you have to find another strategy to help your patient. So upregulation and activation of alternative signal transduction pathway is one of the main proposed mechanism of resistance in endocrine therapy resistant in breast cancer. One of the most important pathway is PI3 kinase AKT/mTOR pathway, and the other pathway is cyclin-dependent kinase, which is a key factor in cell cycle progression. Other proposed mechanism also is loss of receptor, and when you are losing your target treatment will not become effective. Other mechanism of resistance is alteration at the genomic level, and this will change the makeup of your target and make the target treatment less effective. This has been the focus of the research for the past 10 years or so, and the researcher came up with many strategies to overcome estrogen pathway resistance and to make the targeted treatment that we have more effective. One of them is targeting or change the mechanism of targeting the estrogen receptor by itself. Fulvestrant is one of main example for that, by using another method of blocking estrogen pathway. As I said, PI3 kinase AKT/mTOR pathway is also one of the main pathway in development resistance, and we have very successful story with using mTOR pathway inhibitor, everolimus, in combination with endocrine therapy to actually overcome the resistance to aromatase inhibitor and first-line endocrine therapy monotherapy. As Dr. Thill also touched base on using PI3 kinase. Using PI3 kinase is very promising target, and inhibiting that target also came up with a good result, especially in subset of the patient with mutation in PI3 kinase. However, this is not really for clinical use because of the toxicity that's coming from this agent. Many other pathways have been studied also, including RAS pathway, SERT kinase pathway, using proteasome inhibition like Velcade, and even using HER2 receptor inhibition and EGFR inhibition in combination with endocrine therapy. However, most of this study did not show much clinical benefit. So, overcoming endocrine resistance, most likely it's not a simple question that has a simple answer or one approach that will solve and overcome this resistance. However, using more than one approach and modalities to treat the patient maybe is the answer for that. And actually what has been and what's shifting the paradigm in the last 3 or 4 years is introduction of CDK4/6 inhibitor. And actually this is changing the practice in luminal breast cancer. In 2017, we have 3 drugs that are approved as a CDK4/6 inhibitor to be used in advanced post-menopausal breast cancer patients. The first in class was palbociclib from Pfizer, and it was approved in 2015, based on phase 2 study. And the most recent one is ribociclib that was approved earlier this year, and just a few weeks ago, abemaciclib was approved to be used in advanced luminal breast cancer. CDK4/6 inhibitor is a novel mechanism, drug that uses novel mechanism of action by arresting the cell cycle. And as if we think about simply the cancer and the tumor, if we can arrest the cell cycle and decrease the proliferation, this might control the disease. So cyclin D is one of the key factors and one of the main target for direct transcription, that target for estrogen receptor. And blocking the combination of CDK4/6 inhibitor and cyclin D will inhibit the phosphorylation of retinoblastoma, and this will inhibit the release of E2F transcription factor, and eventually this will lead to cell cycle arrest. And actually, this line of research is not something new. Actually, it's extended to the late 90s. However, in early 2000s, the clinical development of this compound fell short basically because of lack of selectivity and because of introduction of pan-CDK inhibitor. In 2009, Richardson and Dennis Solomon, UCLA, observed very interesting observation, which actually that's what led to all the progress that happened in the next few years. Trying this compound, which is the palbociclib, the Pfizer compound, on a cell line that contain all subsets, intrinsic subset type of breast cancer, they observed that this compound is only working on luminal breast cancer, or it's more effective causing anti-tumor growth in the luminal cell line. And this observation actually led quickly to development of therapeutic interventional trials. And the first one was, after the preclinical and early phase trial, was Paloma-1 trial, which is a phase 2 combination of CDK4/6 inhibitor palbociclib with the standard of care letrozole compared to letrozole monotherapy as the standard of care that we are using for treating advanced breast cancer. And as you see here in Kaplan-Meier curve, there is very obvious improvement in progression-free survival. Progression-free survival was the main objective of this study, and it showed about 10 months improvement in progression-free survival with very significant hazard ratio, 0.4. So actually, based on the data from this trial in March 2015, the palbociclib granted accelerated approval, and it was approved also waiting for the more extended phase 3 trial. One of the main questions that came at that time, and it was actually very important when you are talking about improvement in progression-free survival, are those novel agents safe? Are they the toxicity profile is acceptable? And what the early phase study showed is the toxicity profile is not that significant comparing to other targeted agents, and actually it was very tolerable for the patient. The main toxicity for those agents was myelosuppression, especially neutropenia. However, it was asymptomatic neutropenia, even if it's higher grade. Most of the patients will not feel that it was asymptomatic, and it will not cause a lot of morbidity for the patient. So this was a good news for the people who developed the drug, and actually for all oncology community. This led to more extended phase trial, phase 3, and also introduction of new agents. That was studied in phase 3 trial, including ribociclib and palbociclib, compared also to the standard of care in the first-line setting. The data were strong, were reproducible. It showed the same thing in phase 2 trial, and also it was reproducible across the different agents. So it seemed to be a class effect rather than one compound effect. The progression-free survival also was about 10 months, and with very significant hazard ratio of 0.4 to 0.5. Asking the question, does this effect is restricted to one subgroup of the patient, either one demographic group of the patient or one clinical subgroup of the patient? And it seemed to be even across all the trials. The class effect of those drug, it's effective in all subgroup of the patient, regardless what's the grade of their disease, or if they have prior adjuvant therapy or not. Looking also again for toxicity in phase 3 trial. Very interesting finding was it's also the toxicity is a class effect rather than one compound effect. And neutropenia, asymptomatic neutropenia, was the main side effects for those agents. Other minor side effects was only fatigue and nausea. Febrile neutropenia was very tolerable, and it was not that much in rate of 1 to 2% for all patient population in both phase 3 study for palbociclib and ribociclib. Maybe there is a minor toxicity that different from ribociclib and palbociclib, which is increase in the QTC interval that was observed more with ribociclib, and actually it's not observed with palbociclib. So the recommendation even from the company is to observe a QTC interval for the patient in the first 3 to 4 visit, and not to use anything that might increase the QTC interval. However, because of the main objective of the study was progression-free survival, and there is no data about overall survival, it was very important for the researcher to find out, is there a subgroup of the patient who might benefit from those medication? And unfortunately, and this has been studied in many across different trials, there is no predictive biomarker that will tell us which patient might benefit from this novel drugs. So we have to use it now only based on our clinical judgment. Even using Ki-67 or overexpression of cyclin B1 or loss of P16, there is no signal that there is those predictive biomarker can help in our selection to our patients. Because I don't have much time for the rest of my study, I just mentioned something about the third agent, which is abemaciclib. It's more potent CDK4 inhibitor, so we think it might have different indication. The chemical structure for this drug is different, and hopefully this will make us have a different indication for this drug. The data for abemaciclib is also similar to the data for palbociclib and ribociclib. One of very interesting finding from MONARCH 3 was this exploratory subgroup analysis that was done by the investigators. And it showed that patients with more aggressive disease, the patient who has less treatment-free interval, and the patient with visceral disease, they might be more sensitive for abemaciclib. And actually, this information, if we can approve it with a larger number of the patients, it might be practice changing, and it might put abemaciclib first in this race between the three agents. So there's many trials that will come in the recent future, which will give us more information. It will tell us about use of those agents in premenopausal patients. Can we use them in brain metastasis patients also? So at the end, we think that those agents are very promising, and as Dr. Thel say, there is a desperate need for biomarkers to identify the patient and the tumor most likely to benefit, and those who will do well with hormone therapy. Thank you so much.